Hello students, so coming to the last part of chapter number 11, that is biotechnology, principle and processes. In this video class, we will study about the stepwise process of recombinant DNA technology, okay? And in previous class also, we have studied about the tools and about the basic step of recombinant DNA technology. But in this video class, we will discuss in details stepwise processes of recombinant DNA technology one by one, okay? So for recombinant DNA technology, the first step is isolation of genetic matter. That is to extract the DNA from the donor. And here the main challenge is that this DNA need to be extracted in pure form. And DNA is never present in pure form, but still it need to be extracted in pure form. So we will study how this DNA is extracted in pure form, okay? So to obtain the DNA in pure form, we have to break open the cell membrane as well as nuclear membrane, and we have to remove all the unwanted macromolecules. Only then we can obtain DNA in pure form. And another point is that we have to say whether cell wall is present or absent. And if cell wall is present, we have to digest or remove the cell wall. And for this, we can use lysozyme for bacteria, cellulase for plant, chitinase for fungi. Okay. And after this, the treated cell are centrifuge. Here, the cell will be rotated at high speed with the help of a device, and this will result in breaking down of all the membrane and all the macromolecule will be mixed up. Okay. And from this mixture, unwanted macromolecule will be removed with the help of different enzymes. For example, RNA can be removed with the help of ribonuclease, protein can be removed with the help of protease, like that. Carbohydrate will be removed with the help of amylase, and lipid with the help of lipase. Like this, all the unwanted macromolecules can be removed with appropriate enzyme. However, the DNA will remain intact. And after this, the DNA can be precipitated out by the addition of sil ethanol. Like this, we can extract the DNA in pure form. aduna recombinant DNA technology ki step lakanda aham step se kari no kanda isolation of genetic material ne DNA se extract to ni kari form da pure form da important point di DNA do pure form da yoro moi have makadoni protein ga adum bound to lemane aduna sigi DNA se combine to ka pure form da extract to ni have sne on aham da khoi kai se to ni whether cell wall is present or absent yoro maba cell wall do dies on da Auna bacteria dadi cell wall jawbnina it can be digested with the help of lysozyme. Plants cell wall jawbi, so it can be digested with the help of cellulase. Like that, fungi su cell wall jawbnina it can be digested with the help of kanano chitinase. Ade kadogani scientific togani akoina device am sinaraga ngasagi cells rotate togani high speed ta sina kai to honge kanda mondariba membranes in the loina kairani and everything will be mixed up. Loina mix up torneba. ꯑꯗꯨꯒꯤ ꯃꯤꯛꯁꯆꯔꯗꯨꯒꯤ ꯑꯩꯈꯣꯏꯅ ꯀꯩꯗꯧꯒꯅꯤ ꯍꯥꯏ ꯀꯥꯟꯗ ꯑꯟꯋꯥꯟꯇꯦꯠ ꯃꯦꯀ꯭ꯔꯣꯃꯣꯂꯤꯀꯨꯜꯁꯤꯡꯗꯣ ꯔꯤꯃꯨꯚ ꯇꯧꯒꯗꯣꯏꯅꯦꯕ ꯑꯗꯨꯒ DNA ꯈꯛꯇ ꯊꯝꯍꯧꯗꯣꯏꯅꯦꯕ ꯑꯗꯨꯅ ꯑꯟꯋꯥꯟꯇꯦꯠ ꯃꯦꯀ꯭ꯔꯣꯃꯣꯂꯤꯀꯨꯜꯗꯣ ꯀꯃꯥꯏꯅ ꯔꯤꯃꯨꯚ ꯇꯧꯒꯅꯤ ꯍꯥꯏꯔ ꯀꯥꯟꯗ ꯑꯩꯈꯣꯏꯅ ꯑꯦꯟꯖꯥꯏꯝ ꯁꯤꯖꯤꯟꯅꯔꯒ ꯗꯥꯏꯖꯦꯁ ꯇꯧꯍꯟꯕꯅꯦꯕ ꯑꯗꯨꯅ RNA ꯌꯥꯎꯔꯝꯃꯦ RNA ꯗꯥꯏꯖꯦꯁ ꯇꯧꯍꯟꯕ ꯌꯥꯔꯅꯤ ꯋꯤꯠ ꯗ ꯍꯦꯜꯞ ꯑꯣꯐ ꯔꯥꯏꯕꯣꯅ꯭ꯌꯨꯀ꯭ꯂꯤꯌꯁ ꯄ꯭ꯔꯣꯇꯤꯟ ꯌꯥꯎꯕꯗꯣ ꯄ꯭ꯔꯣꯇꯤꯟ ꯆꯥꯊꯣꯛꯍꯜꯂꯅꯤ ꯗꯥꯏꯖꯦꯁ ꯇꯧꯍꯜꯂꯅꯤ ꯋꯤꯠ ꯗ ꯍꯦꯜꯞ ꯑꯣꯐ ꯄ꯭ꯔꯣꯇꯤꯌꯦꯁ ꯀꯥꯔꯕꯣꯍꯥꯏꯗ꯭ꯔꯦꯠ ꯀꯅ ꯋꯤꯠ ꯗ ꯍꯦꯜꯞ ꯑꯣꯐ ꯑꯦꯃꯥꯏꯂꯦꯁ ꯂꯥꯏꯛ ꯗꯦꯠ ꯂꯤꯄꯤड ꯌꯥꯎꯕꯗꯣ with the help of lipase aduna unwanted macromolecules in koi sathoimane sista DNA sathopa ngamba enzyme DNA se koi add toba yade ko adu kadawni ngasagi ethanol se add toksanbani ethanol se add torkanda PPT form token phamdara kaneba adu phamdari poto kanano it is the pure form of DNA like this we can extract the DNA in pure form. So coming to next step, that is cutting of DNA at specific location. We know that DNA will be cut by a specific restriction enzyme at specific location, and after DNA is cut into fragments, the targeted or desired DNA will be selected out through cell electrophoresis. And we also know that with the help of same restriction enzyme, the vector will also be cut so that the donor DNA and vector DNA will produce sticky end, and as they are cut by same enzyme, the two sticky end will be complementary to each other and they can be easily joined together by DNA ligase to form recombinant DNA. For this formation of recombinant DNA, we have discussed earlier, okay, in previous class. But before the desired DNA is inserted into the vector, the target DNA need to be amplified. That is, it need to be produced in large number, and this can be achieved through PCR. So our next step will be PCR. Aduna akwegi biotech process lakanda second step se kaina kanda cutting of DNA at specific location. kanana cut togani harakanda restriction enzyme cut togani ko into smaller fragment or sigi fragment se DNA fragments dagi akoina desired DNA se select toganeba through gel electrophoresis aduga ason side ta same enzyme se sinaraga akoina vector su cut toganeba karigino harakanda same enzyme cut torobani na ngasagi desired DNA plus a donor DNA plus a sigi vector DNA moina produce ba sticky and anido complementary or complementary una kadore they can be joined together by DNA ligase aluke form to gani recombinant DNA form to yadine adubu recombinant DNA form to ding da kari to dakar ta ge har kansta desired DNA to amplify to masinga mamam dakar ta ba adu masinga mandai kai process toroga PCR toroga polymerase chain reaction toroga aduna koi next topic se kana tare PCR polymerase chain reaction, that is PCR. So this PCR was invented by an American biochemist by the name Kerry Mullis in 1984. And this PCR actually it is a method which is used to make millions of copies of specific DNA, or you can say it is the in vitro synthesis of multiple copies of a gene of interest or a specific DNA sequence. Okay. And when we come to the requirement for PCR, two set of primer will be required, and these primer are short stretch of single-stranded DNA which are obtained to 18 nucleotides and which is complementary to the 3' end of the template strand. In addition to this, polymerase is also required, and the polymerase that we are going to use during PCR is called tag polymerase, because the particular polymerase is extracted from a thermostable bacteria, that means which are stable at high temperature, thermostable bacterium by the name Thermus aquaticus. Okay, and in addition to this, we have to supply all the required nucleotides. So these are the requirement for PCR. So PCR da tawdise karino haitaragadi ngasagi akoina target DNA do in vitro outside the body haitara akoina produce tawdani in large number in artificial way aduna DNA duna divide to da dakar tariba post in do koi loina supply to gotene anu kai dakar tarani primer howdona ba primer dakar takne sigi primer se tasina kanano haitaragadi DNA ki short single stranded sequence amani aduga maruina kanada complementary gane kanda ngasagi parent strand oideba DNA do adugi 3' and thereby magi sequence to aduga complementary oideba sigi matakta akoina nucleotide hapkani nucleotide add togani nucleotide kana add togani polymerase na aduitadi polymerase will be required adubu PCR habasidi high temperature da togani na polymerase to it should be stable at high temperature aduna ngasagi PCR da toriba polymerase akoina kanada ge extract toge harakanda bacteria amadagi by the name Thermus aquaticus ar hariba bacteria se high temperature stable of bacteria name aduna monirothoiba polymerase to definitely it will be stable at high temperature mai to dana ngasai PCR to da dakata ba primer ani already tag polymerase nucleotide se akoina loina supply togane so these are the requirement of PCR. The PCR it takes place in three main steps. The first step is denaturation, second step is annealing, and third step is extension. And here in the first step there is denaturation. The targeted DNA will be heated at high temperature at about 94 degrees Celsius for about 2 minutes. And this will result in breaking the hydrogen bond present between the two strands of targeted DNA. And thus the two strands of DNA will separate and DNA will become single-stranded. And this single-stranded DNA will function as template for the synthesis of new strand of DNA. aduna akoigi PCR harise makadoge kanda step omda toganeba aham steps na denaturation ni second steps na annealing third steps na extension have aham steps denaturation se sita togdi se kaina kanda target DNA to hit togadaneba at high temperature kyam high gedamna kanda 45 degrees Celsius swaimu to ko hit toganeba sina kadawani magi hydrogen bonding kairani double stranded DNA do it will become single stranded DNA aduga hariba single stranded DNA sina parent strand template strand ne ektoga maga complementary ba anoga strand ama produce togadeni. Next come to second step, that is annealing, and for this step the reaction temperature should be around 54 to 60 degrees Celsius, and here the primer are added to their complementary sequence on the template strand on the template strand or template DNA, and here the primer should be added towards the 3' end of the template strand, and this primer will serve as a starting point for the synthesis of new DNA strand. Aduna ꯑꯩꯈꯣꯏꯅ ꯁꯦꯀꯦꯟ ꯁ꯭ꯇꯦꯞꯇ ꯂꯥꯛꯇꯣꯏꯁꯦ ꯀꯅꯥꯅ ꯀꯥꯟꯗ ꯑꯦꯅꯤꯂꯤꯡ ꯁꯤꯒꯤ ꯑꯦꯅꯤꯂꯤꯡꯁꯤꯗꯤ ꯑꯍꯥꯟꯕꯒꯤ ꯁ꯭ꯇꯦꯞꯇꯣ ꯑꯗꯨꯗꯒꯤ ꯇꯦꯝꯄꯔꯦꯆꯔ ꯈꯔ ꯂꯋꯔ ꯑꯣꯏꯒꯅꯤ ꯁꯤꯅ ꯄ꯭ꯔꯣꯁꯦꯁꯁꯦ ꯀꯔꯝꯕ ꯇꯦꯝꯄꯔꯦꯆꯔꯗ ꯇꯧꯒꯅꯤ ꯍꯥꯏ ꯀꯥꯟꯗ 54 to 60 ꯗꯤꯒ꯭ꯔꯤ ꯁꯦꯜꯁꯤꯌꯁꯇ ꯑꯗꯨꯒ ꯑꯦꯅꯤꯂꯤꯡꯗ ꯇꯧꯗꯣꯏꯁꯦ ꯀꯔꯤꯅꯣ ꯍꯥꯏ ꯀꯥꯟꯗ ꯑꯩꯈꯣꯏꯅ ꯄ꯭ꯔꯥꯏꯃꯔ ꯉꯁꯥꯏꯒꯤ ꯁꯣꯔꯠ DNA ꯁꯤꯡꯒꯜ ꯁ꯭ꯇ꯭ꯔꯥꯟꯗꯦꯗ DNA ꯑꯩꯈꯣꯏꯅ ꯑꯦꯇꯦꯁ ꯇꯧꯗꯣꯏꯅꯤ ꯀꯅꯥꯗ ꯑꯦꯇꯦꯁ ꯇꯧꯒꯅꯤ ꯍꯥꯏ ꯀꯥꯟꯗ ꯇꯦꯝꯄ꯭ꯂꯦꯠ ꯁ꯭ꯇ꯭ꯔꯥꯟꯗꯨꯒꯤ 3 ꯄ꯭ꯔꯥꯏꯝ ꯑꯦꯟꯗ ꯑꯩꯈꯣꯏꯅ ꯑꯦꯇꯦꯁ ꯇꯧꯒꯅꯤ ꯑꯗꯨꯒ ꯁꯤꯒꯤ ꯄ꯭ꯔꯥꯏꯃꯔꯁꯤꯅ ꯀꯩꯗꯧꯔꯦ ꯁ꯭ꯇꯥꯔꯇꯤꯡ ꯄꯣꯏꯟ ꯑꯣꯏꯗꯣꯏꯅꯦꯕ ꯀꯩꯗꯧꯕꯒꯤ ꯑꯩꯈꯣꯏꯅ ꯑꯅꯧꯕ DNA ꯁ꯭ꯇ꯭ꯔꯥꯟ ꯀꯝꯄ꯭ꯂꯤꯃꯦꯟꯇꯔꯤ ꯑꯣꯏꯕ ꯑꯅꯧꯕ DNA ꯁ꯭ꯇ꯭ꯔꯥꯟ ꯑꯃ ꯄ꯭ꯔꯣꯗ꯭ꯌꯨꯁ ꯇꯧꯕꯒꯤ. Next, come to the last step of PCR, that is elongation. And for this step, the required temperature is around 72 degrees Celsius to 80 degrees Celsius. And in this step, new complementary nucleotides are added towards the 3' end of the primer by tag polymerase. And this polymerase, it is the enzyme which can tolerate very high temperature. Okay, and we also know that in this process, the elongation of new strand will always take place in 5' to 3' direction. Like this, we can produce new DNA. Okay. aduna PCR gi last step se kanano haitaragadi elongation ni sigi elongation su a little bit higher temperature to gadini at around 72 dagina 80 degrees Celsius ta arsa to dise kaina kanda new nucleotide se to gadini kanana polymerase haiba enzyme sina ado ngasagi nucleotide se kadona to gani ha kanda hana laiba primer khoni to primer do adugi 3' and to gani aduga hariba process se high temperature to ganina polymerase to it should be stable at high temperature. Aduna sigi matang sa wen sinatiba polymerase kanano tag polymerase. Thermus aquaticus haiba high temperature stable oiba bacteria do adrelo thopa polymerase tag polymerase to mana toroga anuba strand se produce to deneba. Sumain to dena kadokena anuba DNA se produce to bo yarane. Okay. So the point is that if the process is repeated for about 30 times, then about 3 billion copies of DNA can be produced in a very short period of time. Okay, and like this, gene of interest can be produced in large number through PCR. Aduna sigi same process akoina 30 times kuma akoina repeat over taragadi within a short time katabina 1 billion copy of DNA se produce to yani adui taragadi ngasagi desired DNA haigera gene of interest haigera sigi se akoina large number da produce to yani through PCR. Now come to next step, that is insertion of recombinant DNA into the host cell or organism. So here, before the recombinant DNA is inserted into the host cell must be made competent, and we have studied how to make the host cell competent. Okay, but nowadays by using some technique we can introduce the recombinant DNA directly into the host cell, okay, without making the host cell competent. For this we can use microinjection, we can use bioelastic origin gun, and we can use disarm pathogen. ꯑꯗꯨꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯃꯊꯪ ꯂꯥꯛꯀꯥꯟꯗ ꯑꯩꯈꯣꯏ ꯃꯊꯪꯒꯤ ꯁ꯭ꯇꯦꯞꯁꯦ ꯀꯅꯥ ꯑꯣꯏꯗꯣꯏꯅꯣ ꯍꯥꯏꯔꯀꯥꯟꯗ ꯉꯁꯥꯏꯒꯤ ꯔꯤꯀꯃꯤꯅꯦꯟ DNA ꯁꯦ ꯍꯣꯁꯇ ꯏꯟꯁꯔꯠ ꯇꯧꯗꯣꯏꯅꯦꯕ ꯑꯗꯨꯅ ꯑꯩꯈꯣꯏ ꯈꯪꯈ꯭ꯔꯦ ꯍꯣꯁꯇ ꯏꯟꯁꯔꯠ ꯇꯧꯗ꯭ꯔꯤꯉꯩꯗ ꯍꯣꯁ ꯁꯦꯜꯁꯦ ꯍꯥꯟꯅ ꯀꯝꯄꯤꯇꯦꯟ ꯑꯣꯏꯍꯟꯕ ꯑꯃ ꯆꯪꯉꯦꯕ ꯑꯗꯨꯒ ꯀꯃꯥꯏꯅ ꯀꯝꯄꯤꯇꯦꯟ ꯑꯣꯏꯍꯟꯒꯅꯤ ꯍꯥꯏꯕꯗꯣ ꯑꯩꯈꯣꯏ ꯗꯤꯁꯀꯁ ꯇꯧꯈ꯭ꯔꯕꯅꯤ ꯑꯗꯣ ꯍꯧꯖꯤꯛꯇꯤ ꯑꯅꯧꯕ ꯇꯦꯛꯅꯣꯂꯣꯖꯤ ꯁꯤꯖꯤꯟꯅꯔꯒ ꯍꯣꯁ ꯁꯦꯜꯗꯣ ꯀꯝꯄꯤꯇꯦꯟ ꯑꯣꯏꯍꯟꯗꯕꯤꯗ ꯉꯁꯥꯏꯒꯤ ꯔꯤꯀꯃꯤꯅꯦꯟ DNA ꯁꯨ ꯗꯥꯏꯔꯦꯛ ꯏꯟꯇ꯭ꯔꯣꯗ꯭ꯌꯨꯁ ꯇꯧꯕ ꯌꯥꯔꯅꯦꯕ ꯁꯣ ꯗꯤꯁ ꯏꯁ ꯑꯕꯥꯎꯠ ꯏꯟꯁꯔꯁꯟ ꯑꯣꯐ ꯔꯤꯀꯃꯤꯅꯦꯟ DNA into the host cell. After this, the next step is obtaining the foreign gene product. And here what we have to remember is that the product of each gene is protein, and the protein which are produced through recombinant DNA technology in the host cell is known as recombinant protein. Now, I am simple point ni okay mathangi step se kaino ha kanda ngasagi host ta akoina gene ama hapsan matungda adugi gene dugi product se obtain tawdeneba ar gene amagi protein kanano protein oitaba oitadi hai protein se recombinant DNA technology or biotechnology sungabadina adugi protein do kai kore we call it recombinant protein. And here the point is that if in the lab, okay, we can produce this protein in small quantities. adu akoina large quantity da akoina produce kai tadi we have to use a device by the name bioreactor. Okay, so our next topic will be bioreactor. So to produce the recombinant protein in large quantities, we can use a device by the name bioreactor. Okay, so here this bioreactor, it is a cylindrical container of 100 to 1000 liter where raw material can be converted into specific product. And the most commonly used bioreactor is of stirring tank. Okay, so this stirring tank, it is a cylindrical container with a curved base which will help to facilitate the mixing of the content. And here raw material will be converted into a specific product. And this steering tank has also different system like agitator system, foam control system, oxygen delivery system, temperature control system, pH control system, and sampling port. Okay, so these are the character of bioreactor. aduna sel ai point se kaino kanda nga sai recombinant protein se koina large quantities of produce of heter de koi device am system da ka tare and that device is bioreactor aduga koina commonly use riba bioreactor se karamba type na kanda steering tank ni magi character hai tar di ma se cylindrical oiba container ni of 100 to 1000 liter aduga magi base kitang curve hoigani isna kari kan hale mondeba content sing do mix up to the mating pange aduga sista akoin happy ba raw material sing se magi specific product on thok hane sigi mathakta ngasai steering tanks da tongan tom system am yoromani ko mai thapu tona ba agitator system foam control system oxygen delivery system temperature control system pH control system mai to dana sampling port ana aduga hariba bioreactors na ngasai gi darkar ta pathak se loi to hane ba last ta stegi pay product se obtain to gadene okay. Now come to last step, that is downstream processing. Here after the completion of the biosynthetic phase, that is after the sampling pot which is present in the bioreactor indicate the completion of biosynthetic phase, the product will be separated, okay, and after that the product has to be subjected through a series of process before it is ready for marketing. And here the separated product is purified and then they are formulated with suitable preservatives. So all these process like separation, purification, they are collectively known as downstream processing, okay. And in case of drug, clinical trial will be required before they are released for marketing. So like this we can obtain the product in large quantities. Last step, that is downstream processing. Sida tokti se kaino na kanta ngasagi bioreactor have device ba sampling posina ngasagi product se loire na indicate tor kanda koina ngasagi product se koina extract to thone separate torne separate to budana tana ma se ka don koina purify to gani adu koina preservative ka hapte ni ko store to yana ba sigi whole process we call it downstream processing. aduga drug oiba taragadi kari daka tage kanda clinical trial ama tugum changeba before it is released for marketing koina market ta thadok tingi da ko hana clinical trial ama tugum changeba smai to na koina ngasagi product se obtain to kane. So like this we have come to the end of this class as well as this chapter, and today in this class we have studied about the stepwise processes of recombinant DNA technology and hope you can understand each and every point, and thank you very much for your attention.